Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in the previous lecture, we have discussing about the PCR. So far what we have discussed about the different aspects related to PCR, how the PCR is being developed and how you can be able to perform the polymerase chain reactions. And in the previous lecture, we have also discussed about some of the practical aspect where we have shown you how to set up the reactions, how to make the reaction mixtures and how to perform the PCR, how you can analyze the PCR in the agarose gel electrophoresis. So, now in today's lecture we are going to continue with our discussion about the PCR. So, let us discuss about the PCR. So, as you remember that in our previous lecture we were discussing about the setting up the PCR reactions, and in a PCR reactions you have the few of these components, like you have the template DNA, then you have the primers, you have the DNTPs, and then you have the tag DNA polymerase. So, out of these components, the primers, which are the very important components, and for every target gene you have to synthesize a set of primers. So, the primer is a short DNA stretch that serve as a starting point for the DNA synthesis. In PCR you require the two primers to bind on the each single standard DNA flanking the target sequence. These are called as the forward as well as the reverse primers. So, if you have a gene and you know that the gene is always been notified as 5 prime to 3 prime and the 3 prime to 5 prime. So, the primer which actually is going to bind to this side, because if you remember the DNA synthesis is always occurring in the direction of 5 prime to 3 prime. So, what will happen is the 5 prime of end of the primer is actually going to bind to the 3 prime end of the target gene, and that is how this will continue in this direction. So, this primer is called as the forward primer, whereas the primer which will bind onto the reverse strand is called as the reverse primer. So, this is called as reverse primer, this is called as the forward primers. So, now the question comes how you can be able to design the forward as well as the reverse primers. So, before you design the primers, you have to consider many parameters, and considering those parameters only you can be able to design the primers, because when you design the primer, the purpose is that it should actually going to bind to the target DNA, and that complex is going to be recognized by the tag DNA polymerase, and then it is actually going to initiate the elongation steps, and that is how it is actually going to give you the complete amplifications. So, when you synthesize the primers or when you start designing the primers, first thing what you have to remember is about the PCR primer length. So, the oligonucleotide between 18 to 24 bases is the ideal length, which is long enough for the adequate specificity and the short enough for primer to bind easily to the template at the annealing temperature. So, the primer length of very short primers, like if you have a 12 to 16 base pair primers, it is actually going to lack the specificity, because there is a probability that if you have a very small stretch of DNA, it is actually could find multiple regions within the target DNA, or if you are suppose using the genomic DNA, probably there could be a possibility that this particular primer could bind to multiple locations, and that is how it is actually going to give you the not the desirable product, but it is actually going to give you the non-specific products, or the it may not give you the amplification at all. Whereas, if you give very long stretch of DNA, for stretch of primers, like if you take a primer of 40 to 45 nucleotide long, that will have a problem of secondary structures first of all, because it is a very long. So, it is actually going to form the secondary structures, and that is how it is actually not going to bind to the target DNA. Second, that also has a problem that it actually may, because a small portion of that primer may bind to the multiple places, and it is actually going to give you the no amplification or the undesirable applications, and that is why 18 to 24 base pair is a ideal length where you can have the primer length. The third point is, if you are using a very long primers, it is eventually going to end up in being very costly, because primer synthesis is always being done by the companies, and that is how it actually may end up in, you might be end up in spending lot of money, but at the end it is not going to serve the purpose because the what you can get from 18 to 24, the exactly the same or even worse probably would possible if you are using the 40 or 45 base pair nucleotide long primers. Then you have to also consider the Tm of the primer. So, primer melting temperatures. So, primers with the melting temperature in the range of 50 to 58 degrees Celsius generally gives the best results. The GC content of the sequence gives a fair indication of the primer Tm. The two primers should be paired in such a way that their Tm difference should not be more than 2 degree, otherwise it will result in a poor annealing efficiency. How you can be able to calculate the Tm of a primer? So, if you have a primer which is of less than 14 nucleotide, then what you can do is you can calculate the Tm simply by calculating the number of G and C and the number of A and T. So, Tm is equivalent to 4 degree multiplied by number of G and C in the primer plus 2 degree multiplied by the number of A and T in the primers. Now, primer melting temperature is a very important criteria, because primer melting temperature is only going to decide at what temperature you are actually going to allow the annealing of these temperature, and that is how the 50 to 58 degrees Celsius is being found to be ideal. If you take very high temperature, then it will actually having a problem in terms of annealing, and if you take very low number, then it is actually going to anneal at multiple places in non-specific sites. So, that is why it is actually going to give you the non-specific multiplications. The second point is that the primer melting temperature or the Tm of a primer between the two primer, like the forward primer and the reverse primer, should not have a difference of more than 2 degree, because what will happen is if I have set it at primer annealing temperature of 54 degrees Celsius and one of the primer has a melting temperature of 48, the other one is having a 56. So, what will happen is that the 56 is going to anneal, or 50 is because we are setting up at 54 degrees Celsius. So, the 56 ones will go and bind to the target DNA, it will anneal, but 48 degrees Celsius annealing the Tm is, because this temperature is still bigger than this temperature. So, because of that it will not going to anneal. So, because of that there will be no amplification of your target DNA. So, if you have a primer of less than 14 base pair, you can use this sequence or this particular formula, but if you have a primer length which is more than 13 nucleotide, then what you can do is you can calculate the Tm by 64.9 plus 41 degrees Celsius number of GCs in the primer minus 16.4 divided by n, where n is the number of nucleotide in the primers. Apart from that, you also have to consider, so either of these formula you can use depending on the length of the primers to calculate the Tm of your primer, and that value is actually going to be close between the forward as well as the reverse primer. So, that is why you have to design the primer in such a way that it should have a very close Tm value, so that you can be able to very comfortably use the annealing temperature, so that both the primers will actually go and anneal to the target DNA. Apart from that, you also require the primer annealing temperatures. So, primer annealing temperature, too high primer annealing temperatures provide insufficient primer template hybridizations, resulting in a low PCR yield, whereas too low primer annealing temperature will give you non-specific products caused by a high number of base pair temperatures. And the Tm is indirectly or directly being attached to the Ta values or the primer annealing temperature. For example, how you can be able to calculate the primer annealing temperature, 0.3 into Tm the primer plus 0.7 the primer Tm of the product minus 14.9, where the Tm primer is the melting temperature of the primers, that is the both primers, and the Tm product is the melting temperature of the product. So, these primer annealing temperature is going to be deduced once you calculate the primer melting temperature, and you can be able to calculate the melting temperature of the template as well, because if you give the template sequence into the software, it is actually going to calculate the template melting temperatures and the primer melting temperature as well, and that formula you can use, put it into this the primer annealing temperature, and that is how it is actually going to give you the annealing temperature, and that is the annealing temperature you can put it into the software, into the PCR machine when you are setting up the PCR reactions. Apart from that, you also have to consider the GC content, because primer melting temperature as well as the annealing temperature is depending on to the number of G and C present in the primers, and that is why the percent GC content is also very important. The number of G and C in the primer as a percentage of the total basis should be between the 40 to 60 base pair, 60%, which means you should have a very high amount of G and C content, because if that actually provides the binding of the primer to the template, because that actually is allowing very strong binding of the primer to the template, and that is how it actually provides the sufficient time for the tag DNA polymerase to sit onto this primer template complex, and that is how it actually smoothly can initiate the synthesis. Whereas, if you the GC content is very low, the primer template annealing is going to be very weak, because if you remember, the G is always making 3 base pair with C, whereas the A is always making 2 base pair with T. So, if you have more of A and more of the AT rich sequence, like for example, what will happen in some of the pathogenic organism like malaria, you are actually the primer template hybridization is going to be very weak. So, once the enzyme will sit and try to synthesize the DNA, it could possible that during that process the primer may actually dislodge, or the primer is binding to the template, but the hybridization is very weak. Either of that thing, it is actually eventually going to reduce the efficiency of the enzyme mediated polymerizations, and ultimately it may end up in giving you either the non-specific product or the lesser efficiency of the enzyme. Then you also require the GC clamps. So, as the GC forms a stronger bond than the AT, the number of GC content at the 3 prime of the primer should not be more than 3, otherwise it will result in a non-specific tight binding at region where G and C are abundant. So, GC clamp is very is actually going to give you a strong binding than the AT, and because the GC clamp is, so GC clamp is required. What is mean by GC clamp is that if you have a sequence like this, something like this, so in the target DNA the sequence is going to be like this, okay. So, this is actually going to provide the binding of the primer onto the into the template in a very strong way, and that is how it actually ensure that the enzyme will sit and synthesize. But if you have more of these, then it is actually going to give you the problem, because then the primer template interaction is going to be very strong. Apart from that, when you are designing a primer, you also have to consider the formation of the primer secondary structures. So, primer secondary structure arise as a result of intra or intermolecular interaction between the primer or with the primer with the eventually reduce the yield of amplification, as the availability of single standard primer will be limited for PCR. The various types of the primer secondary structures are as follows: the hairpins. So, hairpins are the loop structure formed by the intramolecular interaction within the primer. Optimally, a 3' end hairpin with a delta G of 0.2 kilocalorie per mole and an internal hairpin with a free energy of -3 kilocalorie per mole is tolerated generally. So, hairpins are like a double standard DNA. So, when the primers have the internal symmetry, or when they are actually having the symmetry, they will come and close together, and that is how it is actually going to give you the hairpin like structures, like one part of the DNA is actually going to fold. So, this is the single standard primer, but it actually going to fold and it will give you a hairpin like structure. So, if this will happen, first of all it is actually going to reduce the single stranded length of the primer, and that so that it will be able to having the interaction with the target DNA or the template DNA. The second is because it having a hairpin like structure. So, if the enzyme is sitting here and trying to do the amplifications, it is actually not going to pass this particular hairpin, and as a result the hairpin is actually going to dislodge the DNA, the DNA polymerase, or it is not going to allow the DNA polymerase to go for the to the elongation phase. As a thumb rule, if you have a hairpin loop which has a delta G of -2 or -3, that is well tolerated, because you are going to have the hairpin which is going to be formed when you are putting the restriction enzyme restriction sites into the primer, and that restriction site is always palindromic. So, that actually is going to give you the hairpin, but that hairpin is good enough for going to be broken up when you are putting into a temperature where they are going to anneal, but that temperature is good enough to break these kind of interactions. Then you are going to have the dimers. A primer dimer is a structure formed by a double stranded structure which is formed by the intermolecular interaction between the two primer. If the interaction is formed between the two homologous or same sense primer, it is called self dimer, whereas if interaction is formed between the two different primers, it is called as the cross dimer. Optimally, a 3' with a delta G of 5 kilocalorie per mole or at an internal site with a 6 kilocalorie is tolerated generally. So, what is mean by dimer is that you have, this is the primer number 1 and this is your primer number 1. So, if it's both the sequence are actually having the some kind of homology or symmetry, then they will actually going to form a double stranded DNA, which means they are no longer be available as a single stranded primers, and that is how the purpose of heading the primers into the reaction is going to be over, right. So, this could be because of that, or it could be like one is making a double stranded DNA with the second strand, which means one either making a pair with 1 or one either making a pair with 2. Either of these things, if either it could be a homodimer or it could be a heterodimer. Whatever the case, it is actually going to sequester the single stranded primers, but you do not have to worry about the dimers if the delta G, which is the free energy of this primer dimer, is in the range of -5 to -6, because that is good enough. That free energy is going to be broken down once you keep these primers at a annealing temperature, because that is good enough, because when you calculate the annealing temperature, you are going to consider all these parameters, and that is how you will keep the annealing temperature in such a way that the primer will actually going to be still be able to anneal with the template, but it will not going to be anneal with the next molecule of the primer to form the dimers. Then you are going to have the repeats and the runs. So, repeats are consecutive occurrence of the dinucleotide, whereas runs are the continuous stretch of single stranded nucleotide. A maximum number of repeats and run accepted is a 4 dinucleotides or the 4 base pair respectively. Then you have the primer template homology. So, the primer should be designed in such a way that there should be no homology within the template other than the target site. This will result in a non-specific binding and the amplifications. So, these are the some of the primer secondary structures what you can see. These are the, so if you have these particular type of sequence like this is 2 primers, then you can have the hairpin loops. So, you can have the like hairpin is being formed, and this but you can see that this hairpin has a delta G of 0.16, and that is good enough when you are actually going to heat up and when you are bringing this parameter to annealing temperatures. Then this is an example of the primer dimer, where one sequence of the primer is making an interaction with the second sequence, whereas what you see is this is actually a non-acceptable primer dimers. Like in this case, what you see is that only this region where only the 3 nucleotides are making an interaction with the nucleotides present in the into the other sequence, and then rest all these sequences are very weak interactions. So, this is actually going to be broken down because you have a delta G which is -2.9, whereas what you see here is actually a very strong binding of involving almost 6 to 7 nucleotides, and what you see is the delta G is very high, which is like minus 9.74. So that's why this primer, when you are synthesizing, this primer is not going to be useful, because it is going to be end up in giving you the primer dimers, and that's how it is actually going to and this particular free energy is very high. So it is actually going to give you the non-specific interactions, or this primer will not give you the amplification at all. So this is just a brief overview about how to design the primers. So to explain you how to utilize the softwares, because when you want to design the primers, you can be able to use the softwares for analysis of the multiple aspects, like whether it is forming a hairpin, whether it is forming a dimers, what is will be the free energy and all that. So people are using the different types of softwares, both for designing the primers as well as for analyzing the primers. So let me take you to my lab, and where the students are going to explain you the different aspects of the primer designing and how you can be able to analyze those sequences to decide whether your primer is bad versus your primer is good, because that is very important, because the primer is the the crust of the whole story. If you are designing a bad primer, you are not going to get the amplification, because that is actually going to be give the starting point for your enzyme to go and sit and start the synthesis. So that's why the primer designing is very crucial and very important to understand.
Hello everyone, in this video I will show you how to design the primers and analyze them. So for designing primers, first you have to identify the region of interest, your region of interest which you want to amplify from any vector or any sequence. So in second step, you have to identify non-cutters. There are various softwares available, but we can use New England Biolabs NEB cutter version 2.0. After identifying non-cutters, you have to select a suitable vector in which you want to integrate this amplified region and suitable restriction sites. You will get suitable restriction sites from non-cutters. After that, you can go for designing forward primer. So for understanding purpose, I gave this sequence. So I am using this sequence. I will use this sequence to design the primers and analyze the primers. So this is the whole sequence, but I don't want to amplify whole region. I want to amplify the letters, the sequence which is highlighted in green. So I want to amplify starting from here to here. So now the question arises, what are the non-cutters? So you want to amplify this region and integrate into another vector. For that, you have to identify which are non-cutting restriction enzymes. So what I will do, I will copy this sequence into NEB cutter and identify what are the non-cutters. So I just copy the sequence, paste here, and I will ask submit. So it will analyze the sequence and give non-cutter. These are the enzymes cutting inside the sequence, but we are interested in which are non-cutters. So that means you can see here non-cutters. So just click here. It will give a number of enzymes which will not cut inside the sequence. So once getting this list, we have to identify in which vector you want to integrate your amplified region. So for that purpose, so I have selected, for easy of understanding, I have selected PET23A vector. So you can see this is the vector map. So this is the 5' side, this is the 3' side. N terminal and this is the C terminal side. N terminal means forward primer, C terminal means reverse primer. So I can use BAM H1 in forward primer and XH1 in reverse primer. This is the detailed map. So I have identified two restriction enzymes, that is BAM H1 and XH1. So I can use these enzymes in forward primer and reverse primer. So after identifying restriction enzymes and the vector, we will go for designing forward primer. So I will take this sequence. I want to amplify from here to here, so I will copy this sequence here. So for designing forward primer, it is very easy. You have to take the sequence whatever you are getting up to 15 to 20 bases, you can take as it is. So if you want to insert a restriction enzyme, suppose I want to insert a restriction enzyme, this is the sequence as it is given from this this whole sequence. So I want to insert a restriction enzyme, that is BAM H1. So this is the sequence for BAM H1. Here it cuts, so I can use this sequence here. So this is our restriction enzyme. Here it will cut. So we cannot simply give like this. So there should be some more bases, extra bases we have to add in the 5' side. So I will use so this sequence I will use. So now this is 5' to 3' side. So this is our forward primer is ready. So after designing this forward primer, we have to analyze this sequence, so this primer. So what I will do is I just copy this sequence and I will use Oligo Analyzer software, which is specially designed for this purpose only. I will paste the sequence, just ask analyze. So here also you can see there are so many options are there, like you can analyze hairpin loop, self dimer, heterodimer. So these are the general details. What is the length and GC content, melting temperature, molecular weight. So these are normal details. I will go for hairpin loop. Is there any hairpin loops? So we can see there are number of hairpin loops. We can see different structures predicted by the software. So if you want to explore this thing, you can explore only two bases it is forming, and the delta G value is minus 0.43 kilocalorie per mole. So this is fine. Up to minus 10 kilocalorie per mole is fine. Those hairpin loops broken during the amplification process, but above minus 10 kilocalorie per mole cannot be broken. So in that case, what we will do, either we redesign the primers or we will add 5% 1% B10 or 5% DMSO. These are these chemicals disrupt these loops so that the amplification will be fine. So next I will analyze for self dimer. Is there any self dimers, and what is the maximum delta G? So this is forming continuously 5 bases. It is because of the restriction sites. So those are restriction site. Those homodimers forming due to restriction site can be broken. There is no issue. But other than that, this is also because of restriction site. But other than that, we have to look carefully. So is there any continuously 4 or 5 bases forming this homodimer? Then it is very difficult. These interactions can be broken easily. So here some of the consecutive base pairs are there. These are very weak interactions, so they can be broken. So other than that, there is no significant self dimers. So this sequence can be used. And for heterodimer, predicting heterodimer, you need a complementary sequence with reverse primer, like reverse primer you need. So that we will discuss later on. So we got our forward primer here. So it is very easy to generate forward primer. But in case of reverse primer, it is somewhat difficult, because not in terms of predicting things, it is somewhat tricky. So what I'm saying is, here we have sequence. So in case of forward primer, we just take an as it is sequence, 15 to 20 bases as it is from sequence itself. But here we have to take complementary sequence, not 3 prime to 5 prime or 5 prime to 3 prime sequence. We have to take complementary to this one. Say this is the sequence we got from here. So what is the complementary to this one? So just I will add here. So this is the complementary to this particular sequence. So as you can see, this is we have to keep from this direction, 5 prime to 3 prime. So I will take like this. So what we have to do is, we want to insert a restriction site here. So we can insert a restriction site here directly. So in reverse primer, we wanted to insert XH1 site. So this is the restriction site. As usual, we can use we have to insert T here. So this is the restriction site we added. We can add flanking regions in between, flanking bases before this restriction site. So now we got our reverse primer. So we have to go through same procedure like what I have shown in case of forward primer. So just I will copy paste here and analyze the reverse primer. So is there any hairpin loops? Only one hairpin loop, that is within the range of delta G, so there is no issue. And self dimer, so we can see here continuously 4 bases are forming. In this case we have to either change the sequence or remove some of the bases. We can ignore those restriction, those dimers forming through restriction site. So next, heterodimer we have to analyze. For heterodimer we need forward primer. Just copy, paste here and calculate. It will give is there any heterodimers. This is because of restriction site. This is also because of restriction site. This can be broken. Those which are at the end of the sequence, they can be broken, but which is in middle, if you, those bases are middle, it is very hard to disrupt those interactions, and our amplification will be not good. So there is no amplification literally. Other kind of interactions will be broken easily. These are weak interactions. So this is how we can prepare, design the primers and analyze the primers. We have done all these processes for designing forward and reverse primers, but instead of doing manually, we can do it online. We just have to submit the sequence and it will return the forward and reverse primers. These are some of the tools available online for freely, but there are commercial tools also available like Algo 7 Vector NTA Primer Firmware. So if you interested in these softwares, or you can just go through these sites and submit your sequence, you will get your primers.
So in this particular demo, the students have discussed different aspects related to the primer designing, and I hope you might have understood many aspects where the precautions what you have to take and the what are the things you should avoid while you are designing the primers. So this is like a the classical example where we have synthesized 2 primers in the reverse primer as well as the forward primers. So when you want to synthesize the forward primers, what you have to do is, and this is a primer what we have synthesized for the cloning of the particular gene. So the cloning primers are different from the sequencing primer. So in the cloning primer, what you have to do is, the composition is very important, that you take few nucleotides which actually going to be only for a site for the enzyme to sit, and then you are going to put the restriction site, and then you are going to put the complementary sequence, like the sequence what is been identical to the initial site, like this one, right. So, the forward primer, synthesis of forward primer is very simple, because you can just take the 5 prime end sequence, you add the restriction site on this side, and then you add some more sequences, and that is ready for the forward primers. Whereas in the reverse primers, what you have to do is you have to just take this sequence, you make it reverse, and then you add the reverse the restriction sites, and then you can be able to add some more sequences, so that it will provide the docking site for the enzyme to sit. So that because ultimately you are going to use these primers for making a gene, and then you are going to digest this with the restriction enzymes. So, these are the additional sequences what you are putting, so that the restriction sites are going to sit in on this enzyme onto this particular target amplified DNA, and could be able to chew up. There are multiple things what people might, what the student might have discussed, how to analyze and how to verify and all that. So, this is all about the primer designing and how you can be able to synthesize or design the primers utilizing the different softwares, and I hope you might have understood the whole process, and it will actually going to help you in designing a the primers in your lab. And with this, I would like to conclude my lecture here. In the subsequent lecture, we are going to discuss some more applications of the PCR. Thank you.